Okay, we're in for an exciting session. Just for those, I don't know if there's anyone on who may have applied for the Hope Fellowships, but that review process is currently getting underway, so that'll hopefully we'll get the results out in a timely manner. Well, we will. And we were just saying that we've had an excellent response to the Create weekend in November. In fact, our accommodation is maxed out, so that it looks like that's going to be a very successful session. Now, I might hand over to Romy to perhaps give us a little bit of background and introduce Monica for today's talk. So over to you, Romy.
Thanks very much, Paul, and thanks everyone for joining online. It gives me great pleasure to introduce Monica today. I think some of you would know her already from meeting her at the Create weekends and TSNZ perhaps, and some of you might have seen her at the ERS as well. So Monica has been very closely co-supervising with me Archana Gaikwad. Archana was the PhD student who started off the work on vascular remodeling in IPF, and then looking at the endo-EMT changes as a potential contributor to vascular remodeling, especially arterial remodeling in IPF patients. So she's just graduated and she's now officially become Dr. Archana Gaikwad. So she's happy. She graduated this year. So she started off with vascular remodeling work, which got published earlier. Then recently, early this year, she published the endo-EMT work looking into that. And currently, we got data which is under review with one of the journals looking at drivers of endo-EMT, and that's currently being reviewed. So that's where we are with that. And Monica is going to give an overview of where we are with that work and how we're going forward. And apart from that, she's also, as her own interest, she's looking into the epithelial changes in IPF as well, also looking at type 2 pneumocytes. So that work is in preliminary stages at the moment. So that's work in progress. She's quantifying the tissue, analyzing, and trying to make sense out of it. But we'll present whatever she's up to so far. So thank you, Monica. Thanks, Paul, and everyone. And I'll hand over to Monica to take us through the talk. Thank you.
Thank you, Romy, and hi, everyone. I will share my screen. Just give me a second. Oops, wrong one. Can everyone see the screen? Yeah. Okay. So, as Romy mentioned, I'm going to present some our PhD student Archana's work, and also my work on mesenchymal transition in IPF. And first of all, we know IPF is a irreversible fatal fibrotic interstitial lung disease with 2 to 4 years of survival, and with the stimuli such as cigarette, and it's under the umbrella of the interstitial lung disease, and the idiopathic, which means is unknown, so we don't know. Although now we know some risk factors and the reason for the disease, but for the actual disease, the cause of the IPF, we still need a lot of research on that. And we know there are some risk factors such as the smoking, which is considered the major risk factor, can cause the fibrosis and the airway remodeling, and leading to the fibrosis forming in the airway. And also there are some other factors such as environmental pollution and the genetic changes, and also infections can also cause the fibrosis in the lung. And then there are some pathologies where we know that for IPF or causing the fibrosis, as you can see in this image, on the left side is a normal lung tissue. It looks like in the parenchyma, especially, we can clearly see the the space of the alveolar region and the normal, the normal airways and arteries next to it. And in contrast, on the right side is the IPF tissue, and we can see there the normal alveolar structure was completely replaced by the fibrosis tissue, and also forming the typical structure of IPF is called honeycomb beam structure. And also we can see this artery is very compared to the normal one, the very thick of the have a very thick arterioles. Can you see the point? Sorry. Yeah, so yeah, this is better. So this is the, for example, this is the artery in the IPF tissue. We can see very thick arteriole, and also the parenchyma change. And so we know there are some cell type can change to form the fibroblasts. For example, in the airway, we can the epithelial cells and type 2 pneumocytes, they can, under the EMT, which is called epithelial mesenchymal transition, that process, they can form as the fibroblast. And then also in the artery, it also can under the mesenchymal transition process. So the endothelial cells can change and translate into this the mesenchymal phenotype, which also can lead to the fibrosis. And firstly, we're looking at, and we know that pulmonary arterial hypertension is one of the complications in IPF patients. It affects the patient's survival. So we are, firstly, we're looking at this, the artery, the pulmonary artery in IPF. So pulmonary arterial hypertension is a type of high blood pressure that affect the arteries in the lung and the right side of the heart. And there are 32 to 84% of IPF patients suffer from this pulmonary hypertension. And let's look at some images. So we use Movite pentachrome spectral staining, look at the arteries in the normal and the normal just non-IPF pulmonary hypertension patient's tissue. As you can see on the left, in the normal arteries, the artery wall is very thin, and the lumen have a proper lumen size. But in contrast, in the pulmonary hypertension, the artery wall becomes very thick, and the lumen reduces the size of the lumen. And then we did the same staining in IPF tissue, and we see the similar structure of the artery remodeling, the similar images to the pulmonary hypertension. As you can see, the wall is increased, and also the lumen size is quite irregular, and a lot of elastin deposition. And so based on this staining, we are hypothesizing that the endothelial cells may be transformed into the fibrosis that's caused this remodeling. So we want to identify whether the pulmonary artery remodeling is happening in IPF. So we conducted our first study, is looking at the size-based quantitative analysis in pulmonary artery in IPF patients. So we have 11 normal control and 13 IPF resect lung tissue, and we did the Movite pentachrome staining. The first thing we we measure the artery size. So based on the external lens and the luminal lens, to classify the artery size into different groups. And the reason we use a MOVAD pentachrome staining is it's very colorful and it can stain 5 different regions at one go. And each part of the component of the artery is very clearly. For example, the elastin is showing the black and the muscle layer is in red. So we can see what's the component in the remodeled artery. So here, based on the size classification, as you can see, the smaller size and the top panel is the normal control and the bottom panel is the IPF. And in different size, we clearly can see the artery is remodeled. And then we did the quantification of the artery numbers in the IPF tissue and the normal compared to the normal controls. As you can see, the numbers of the artery was significantly reduced in IPF patient compared to the normal control. Yeah. And then after the classification, we want to measure the thickness of the artery. So we measured the total artery thickness is from the lumen side to the external of the adventitial layer. And also we measured the individual layers of the artery, including intimal layer, medial layer, and the adventitial layers. And we found significant increase in thickness across all the size in IPF compared to the normal control. And then we look into individual layers and across all the size. As you can see here, in the from medium to large size of artery, all the layers, intima, media, and adventitial layers are all thickened compared to in IPF compared to the normal control. And in the smaller size, the intima was significantly increased. So here we the message is the intima thickness was significantly increased. So yeah. So another component in the artery is the elastin deposition. Then we quantified the elastin deposition in the arteries as well. So we measured the thickness and we quantified the elastin deposition. So as you can see, across all the size of the artery, the elastin deposition in IPF is from the medium to large size, the elastin deposition in IPF was significantly increased compared to the normal control. And the fragmented elastin also observed across all the artery walls. Oops. So this study has been published in 2022 in ERJ Open Research. So please refer to this paper for more details. And now we know that the pulmonary artery remodeling is in IPF. And what's the next question is what's the mechanism driving the remodeling? So we look into the endothelial mesenchymal transition. We use the endothelial, the endo-MT markers to evaluate whether this transition is active or related to the remodeling. So first of all, briefly introduce give some background of the markers. So VE cadherin is the endothelial marker we used in this study. So endothelial VE cadherin is an endothelial specific adhesion molecules located into the junction. So it binds the endothelial cells together. The mesenchymal markers, N-cadherin, is also a junctional connecting protein, but it more represents the mesenchymal phenotype cells. And the Vimentin and S100 are both mesenchymal markers. So they are showing the fibrosis. The fibrotic cells and the mesenchymal cells. So we use the immunohistochemistry staining to look at these markers expression in the IPF patient's tissue and also in the pulmonary artery. So first of all, let's look at some staining image. As you can see, the VE cadherin this is an artery in the IPF tissue, and we can see the VE cadherin is expressed in the endothelial cells and mostly expressed in the cytoplasmic. And also N-cadherin is quite positive in the endothelial cells and the layers of the artery, and the Vimentin S100 as well. So these mesenchymal markers are quite positive in the IPF tissue. So we quantified these markers expression. First of all, because VE cadherin is supposed to be the junctional marker, so they should express in the junctional. So we look at the junctional positive and the cytoplasmic negative cells, and we can see in normal control, the VE cadherin is mostly junctional positive, and but compared to IPF is less junctional. But in contrast, we found that in cytoplasmic positive of VE cadherin is more in the IPF and less cytoplasmic positive in the normal control, which means the VE cadherin is transformed from the junctional into the cytoplasmic. And then the N-cadherin we also find is quite significantly higher in IPF across all the size range of the pulmonary artery compared to the normal control. And similarly, Vimentin and S100 for showing the same increase in the IPF. And as mentioned earlier, the VE cadherin and the N-cadherin are the junctional markers. So we look into the junctional expression and the cytoplasmic expression. So the top panel is showing the VE cadherin junctional versus positive versus cytoplasmic positive. So this showing the IPF is more junctional. In the IPF, the VE cadherin is more cytoplasmic positive, and junctional is more in the normal control. And similarly, in contrast, the N-cadherin, the junctional positive is less in normal control and also less in positive, the cytoplasmic, but it's very high in both junctional and the cytoplasmic in IPF, which means the NDMT is active. So the VE cadherin translocate into the cytoplasmic and also gain the endothelial cells, also gain the mesenchymal phenotype because the N-cadherin is increased. So then we did some correlation with the artery, the NDMT markers with the artery, pulmonary artery remodeling factors. So first of all, we did the correlation with the total thickness. So we can see that the N-cadherin and the Vimentin have a strong positive correlation with the total thickness, and also these two markers have a strong positive correlation with the elastin deposition. So, and this study have been published early this year, and also in ERJ. Please refer for the more details. So now we know that pulmonary artery remodeling is in IPF, is active in IPF, and endo-MT is the mechanism behind this remodeling. So now the new question is, what drives the endo-MT activities? What's the mechanism behind this? So we, similar to the EMT, so similar to the epithelial mesenchymal transition, there are several signaling pathways are involved into the endo-MT process. So TGF SMAT signaling pathway and Wnt pathway commonly relate to the mesenchymal transition. So this study, we are using these signaling biomarkers, TGF-beta, phosphate SMAD2/3, SMAD7, and beta-catenin, to look at whether this signaling pathway is active and relate to the endo-MT process. And then briefly, in TGF-beta signaling pathway, TGF-beta activate binding to the TGF-beta receptor and activates the phosphorylation of SMAD2 and 3. And the phosphorylated SMAD2/3 will bind to the SMAD4 and translocate it into the nucleus and binding to the DNA factors and involve into some transformation, affect these markers, these proteins formation. And the Wnt pathway is related to the beta-catenin, which also will relocate into the nucleus and involve into the DNA transform. So these two signaling pathway, and then we look at into these markers in the IPF. So similarly, we did this quantification of each markers in the pulmonary arteries. So first of all, the TGF-beta1, we can see there are significant increase TGF-beta1 expression in IPF pulmonary arteries compared to the normal, and then across all the size ranges. And then we look at into individual layers and in each size and we can see especially in the intima, it have a significant TGF-beta expression across all the size ranges. And the phosphorate SMAD2/3 expression also have a very high expression across all the sites of the in IPF compared to normal, and in all the layers have a high expression in IPF. And beta-catenin also showed similar to the TGF-beta. We can see it's across all the size range except the large one have a significant high beta-catenin expression, and but in intima layer, even the large size of the artery have the very significant beta-catenin expression in IPF compared to normal control. And interestingly, we find the SMAD7 expression also highly expressed in all the size range and all layers of the artery size, artery. So, yeah, so we see the markers are increased, and what's the correlation with the pulmonary artery thickness? We did some correlation. We can see TGF-beta1 and the beta-catenin have a strong positive correlation with the pulmonary artery thickness, total thickness, and also the intima layer thickness. So in the last slide, we saw that these markers higher in intima. So, and it have a So and this slide also showing the intima have the thickness have the correlation with this markers expression. And then how about the correlation with NMT markers? So we found that first of all, we found that TGF-beta1 have the positive correlation with Vimentin, N-cadherin, and also the V-cadherin cytoplasmic expression. And similarly, with beta-cadherin also have the positive correlations. So this tells us that these signaling pathway markers are correlated with the pulmonary artery remodeling at the thickness and also the endo-MT markers. And then lastly, we look at the correlation with DLCO, the lung function. So we found TGF-beta in total artery TGF-beta1 and the beta-cadherin have negative correlation with DLCO, and also the intima expression have negative correlation trend with DLCO. Here, although it's the p-value is not significant, but because the sample size is less, so we couldn't see the significance, but we can see the negative correlation trend with the DLCO. So to sum up this study, so the significantly increased the TGF-beta, p-SMAD2/3, beta-cadherin, and the SMAD7 indicate that the TGF-beta and the Wnt signaling pathway is involved into the pulmonary artery remodeling and is driving the endo-MT activities. And this study have currently is under journal review. So hopefully we can get some feedbacks and yeah. And okay, so that's the part of the Archana's study. And I'm also, since we know that the mesenchymal transition is in IPF and relate to the pulmonary artery, but that transition is not only limited in the artery remodeling. And we can see, we saw some images that in the IPF, fibrosis is everywhere. It's also involved into the airway. So my study is looking at the epithelial mesenchymal transition in the IPF patient and also LEM patients. So first of all, I will introduce what is LEM. So LEM is characterized by hyperproliferation of abnormal mesenchymal smooth muscle-like cells, resulting forming the multiple lung cysts and the cyst lesions in the parenchyma. So the smooth muscle cells of the lungs start to overgrow and resulting the formation of multiple cysts and causing the lung tissue fibrosis and the scarring, and which is and this disease majorly found in women, and the life it's about 10 years after diagnosis. So we previously we discussed about the endothelial mesenchymal transition. So here is epithelial mesenchymal transition, which is similar. So the epithelial cells are losing the junctional connection. These markers, for example, E-cadherin, Z01 cytokeratins, and the cells are slowly gain the mesenchymal phenotype, and finally gain these mesenchymal markers and present as a mesenchymal cells. There are 3 types of the EMT epithelial mesenchymal transition. Type 1 is is organ development, so it's a normal process. So in our body, EMT is happening, and the type 2 is related to the wound healing and the organ fibrosis, and also type 3 is related to the malignance and the angiogenesis. So we are so for the IPF fibrosis, we are looking at maybe type 2 and type 3 EMT. So similar normal control and the IPF cohort were used in this study, and we also include the LAM cohort. Although we only have 6 patients, and some of them have the comorbidities such as asthma, COPD. Yeah. So similar IHC method were used, and we use the E-cadherin, N-cadherin Viminton S104 for the staining, and the E-cadherin is the epithelial markers for epithelial junctional, present the epithelial junctional. So how we did the quantification? So first of all, for E-cadherin and N-cadherin, we select the positive, so the brown color indicates the positive. So we select this color and select the epithelial region, and we got the percentage of the positive expression of this marker in the epithelial region. And we, for the Viminton and S104, we count the positive cells individually and normalized with the reticular basement membrane, the yellow line, the length. So we average that, got the cells per millimeter of the RBM length. So here are some data. So first of all, the E-cadherin expression in epithelium. As you can see the images in normal, we can see very strong dark brown color of the E-cadherin in the junctional of each epithelial cells. Although we can see some positivity in the IPF and the LAM, but the percentage or the quantification is lower in IPF and the LAM. So which means in IPF and the LAM, the epithelial cells gradually lose the connective markers, and meanwhile it gained the mesenchymal markers. As you can see here, in normal, N-cadherin is quite negative, and in IPF and the LAM is very strong, and especially in LAM, the N-cadherin is very strongly present in the junctional and the cytoplasmic. And then Viminton in the epithelium, similarly, we find negative in the normal controls, and a very strong Viminton expression in the IPF, and maybe slightly can see this few dark brown positive Viminton cells in the epithelium, but it's quite less compared to the IPF. And then the S104, similarly negative in normal control, and quite positive in LAM versus IPF. And then we quantified the Viminton and the S104 expression in the alveolar area, mostly in the type 2 pneumocytes. So first of all, we quantify the type 2 total type 2 pneumocyte in the alveolar area, and we use the type 2 pneumocyte antibody to get the quantification of the type 2 pneumocytes. So we know the cells is type 2 pneumocyte. Type 2 pneumocyte. Then we quantify the Viminton and the S104 in the positive in the type 2 pneumocytes. As you can see, both in Viminton is significantly high in IPF, and S100 higher in IPF and LEM compared to the normal control. So these are just it's very early stage of the very preliminary data. The analysis is still going on, but so far we found that the EMT was E-cadherin was reduced in IPF and the LEM compared to the normal control, and whereas the mesenchymal markers are all significantly higher in the epithelium in IPF and the LEM compared to normal control. And also these mesenchymal markers are markedly increased in type 2 pneumocytes in these two diseases compared to the normal control. So the decline in epithelial marker and increased in mesenchymal marker indicate that EMT is active in both LEM and IPF patients. So there are some limitations. Although LEM was preliminary diagnosis, but as mentioned earlier, some of the patients have the comorbidities, asthma, COPD, and we know that, especially COPD, we know that EMT is active in the COPD. So these comorbidities can maybe attribute to the EMT activities as well, or have some impact on EMT activities. So further analysis need to be done. And also the IPF tissue is from the late stage of the disease. So some of the patients have very limited number of the small airways and the alveolar structure. A lot of the alveolar region were very fibrotic. So it's really difficult to get a proper alveolar region to quantify the type 2 pneumocytes change. But we will do, in future, we will do some further quantification. For example, the fibrotic area and the non-fibrotic area. So we will know how the EMT activities relate to that. And also further analyze the subepithelial layers of the airway. So that will give us a more clear indication that EMT, whether EMT is related to the airway remodeling, and also the correlation relate to the lung function, lung parameters. So here is I would like to acknowledge our RTRG group, and especially Romy and Matthew, giving their support, and Archana did a really good job on the pulmonary artery remodeling in IPF. Is her PhD give this study a very good data on that. And also all the clinicians, collaborators, and Jade and Glenn provides the IPF tissue for us to do the research. And lastly, I would like to share some photos when I was at ERS. So, and also thanks to Romy, I got this opportunity to present a few of our group study. One is the signaling pathway in pulmonary artery remodeling in IPF, and also my EMT study in COPD, and got poster presentation at ERS as well. And also another PhD student, Surajit Day, his study on the inflammatory cell proliferation in asthma COPD overlap. So, and also the scientists and the clinicians at the conference, and it's very good experience. And hopefully next year we can have some other new data in Vienna. And also some sightseeing at Milan. So I stay in Milan for a few days. So walk a lot and looking around, and also enjoy the very nice food. And yeah, it's great experience. And thank you to RTRG and the CRE Fellow. Yeah, that's it. Thank you.
So look, thanks very much, Monica. That was fantastic and very interesting presentation there. One thought that occurred to me, and I'm sure you've thought of it too. I mean, from a clinical perspective, one of the problems of dealing with pulmonary hypertension in secondary to lung disease, and IPF being one of them, is that the typical PAH-specific drugs either don't work, or in the case of endothelin receptor antagonists, sometimes seem to actually make it worse. I was wondering if you might be able to comment on what your findings are, and what the implications of your findings might be for future treatments that deal with this vascular component of the disease. And I guess there had been a sense that the vascular part was all just secondary to destruction and hypoxia, but I think it's moving away from that to a consideration that it's more intrinsic vascular disease as part of the process. Is that what you think?
Yeah, so for our study, I think we try to find some mechanism related to what cause the pulmonary artery remodeling. So from that point, maybe some we can stop for example, in our study, we found the endo-MT is related to the remodeling. So some maybe targeted to the endo-MT, those markers or the signaling pathway, we can stop at inhibit at that stage. So maybe some new drug can targeting to those signaling pathway or the protein inhibitor that may help slow down or prevent the remodeling, then further develop to hypertension, pulmonary hypertension.
Well, I mean, one of the newer agents in PAH is the sotatercept, which is the TGF-beta pathway inhibitor.
Yeah.
Are you aware of have there been any studies of that in models? Someone's on mute. That might be Nicole. The use of sotatercept in I'm just unaware whether it's been tried in bleomycin models or other fibrosis models. Do you know if there's any work in that area?
I'm not sure that area at the moment, but we are also interested in looking at into the cell level as well, like what you mentioned. For we can also at the ERS we talk. I talked to some very nice scientists. So they have the cell model, in vitro model, which we can stimulate the cells and create the fibrosis, or TGF-beta stimulates the cells and cause the fibrosis transforming. Then we can introduce the treatment or the molecular weight for the drug, maybe. So we can do that in vitro model first, and then to further expand our theory or hypothesis. Yeah.
Thanks, Paul. I think that's a good suggestion. We can certainly look into this. I think that's a great idea.
Steve, you can use either just raise your hand or use the raised hand function on the computer there. But Steve, go ahead.
It worked. Just raising the hand. Thanks, Monica. That was a lovely talk. Just a question with regard to the signaling pathways that lead to the endo-MT. You've talked about a couple of them and how they link in. There's lots of different pathways, and one of the key ones that have been identified recently involved in EMT type processes is the mTOR pathway, and there are drugs available for that. Have you looked at mTOR? And secondly, have you thought about going into animal models? Because there's only so much you can do with human cells. Yes, you can look at signaling in vitro and stimulate them, etc. But it'd be interesting if you could get into an animal model if there's a replication. I don't know how similar the glio model and the vasculature and the glio model is, for example, compared to the IPF, but could you go into an animal model, and do you know much about mTOR?
For that pathway, I haven't looked into that yet, but it's a good suggestion. We can, because this study is only for 3 years, and this is AIM-3, so we have time limitation for that another signaling pathway, but definitely in future, maybe I can continue that work and look into different signaling pathway, and definitely that pathway will be look. And also the animal model, that part I'm not sure. Maybe Romy can give some
We'll
comments.
probably chat to Jane about that. We had some discussions with Jane in the past, and I think probably it's time to chat again, Jane, at some stage. Maybe at the Create Weekend.
Yeah.
But good suggestion, Steve. Yeah. So we haven't looked at the mTOR pathway. We started some discussions with Jane in the past to look at the precision cut slices as well, and see if we can explore the NDMT pathway, see what's going up and down, and then take from there into the next stage. And as you said, it's limited you can do with human. And I think the next stage is yes, it is active. The pathology seems to be active and contributing to remodeling. Now, these are the drivers. Can we inhibit them using animal models? So yeah, no, thank you. That's the next stage we're sort of looking into.
Any other questions? Not at the moment. One thought that crossed my mind as you were showing the lamb data, and this is going to reflect my ignorance. The S100 seemed to be particularly up in lamb. What is that actually doing, or is it just a bystander?
That's a very good question. Thanks, Paul. Because now I just done the first step of the quantification of each markers, but what's the meaning behind that and what's why it's high, I haven't figured out. Maybe more after the further analyze, maybe in the subepithelial and all the analyze finish, and maybe looking to because LAM is also only seen in women, though, so the hormone estrogen maybe have some effect in LAM or in S100 for expression as well. Because in early last year, I did some ACE2 TMPRSS, those COVID markers in IPF and the LAM patients as well. And we've also find significantly high of those markers expression in LAM, and that may be due to the estrogen, the hormone effect that increase the expression of those markers. But so I assume similarly situation here with S104, but I need to look into deep why it's higher in LAM particularly.
Okay, all right. Thank you. I think you mentioned BMPs, I think, as well. Is that right? Bone morphogenetic protein markers. Have you been looking at that as well or not?
No, we
Okay.
haven't looked.
Because we, as you may recall, we did some work on looking at that as a sort of counter to the TGF-beta pathway, and certainly seemed to work pretty well in the monocrotaline pulmonary hypertension model.
Mm-hmm.
We had hoped to try it in a bleomycin model, but we had various technical difficulties with the model. So I still think there may be something in looking at that axis as well, because we did have some in vitro. We were, as you say, stimulating cells with TGF-beta and then exogenously upregulating BMPs, either with BMP itself or a virus, and that also seemed to have an impact. I still think there might be something in that one day if we can tease that out. Any other questions or comments? Everyone's overwhelmed by your data, I think, and congratulations on those.
Thank you very much.
ERS. I certainly enjoyed Milan. It was fabulous, wasn't it?
Yeah.
Look, if that's it for now, then I'm happy to wind it up there. I'd certainly be interested to hear how everything's going in your lab. It's a particularly interesting area. And now, Kate, our next meeting is, we got a date for that? I'll put you on the spot now, haven't I? And you're on mute.
Yeah. So it's the 20-something of October. It is it isn't. It's the 17th of October.
18th of October.
And yeah, and I think it's Mariana. No, it's not. It's Alexandra Brown from University of Newcastle.
All right then. Okay. Well, there'll be further notices and reminders coming out for that, of course. And meanwhile, carry on. Good luck with all your grant applications.
Actually, I just told you a fib. That was the November one. Sorry.
Oh.
So it's the 20th of October, and it is Mariana Hoffman.
Okay. All right. Good luck with all your current and future grant applications, and I hope you're all looking forward to the Create long weekend. I think that should be great. I'll leave it at there. Thanks.
Thank
Thanks,
you.
everyone. Cheers.
Bye now.
Thank you, everyone. Bye.